Hi, my name is Lex Kalsert, and I'm the Director of Research Development at the National PKU Alliance. As part of our mission to educate, we have produced a series of short videos to help people with PKU to understand the rapidly emerging field of gene therapy. All new medical treatments carry risks, and not all risks can be easily foreseen. In the U.S., there are many government and research groups to help ensure the safety of individual patients and the public at large. Because the most common delivery mechanism for gene therapy are modified viruses, gene therapy carries additional risks not associated with other types of medicines. These risks include unwanted immune response. Your body's immune system may see the viral vector as an infection and mount an immune response. While normally this is a healthy response, in some cases it can result in inflammation, and in rare cases it can become so severe as to cause organ failure. Scientists have chosen viruses with minimal risk of overactivating the immune response, thus minimizing the risk but not completely eliminating the risk. Targeting the wrong cells. One important role of the vector is to deliver the replacement gene to the specific cells in the body that will benefit from the gene therapy and avoid other cells that will not benefit or possibly even be harmed by gene therapy. In general, these modified viruses are very good at targeting specific cells and avoiding others. Scientists have selected viruses for use as vectors that are known to target specific cells, thus minimizing the risk but not completely eliminating the risk. Infections caused by the virus. Viruses are potent infectious agents. Today, we have very good knowledge and understanding of how viruses work so that scientists can engineer them to minimize the potential for traditional viral infection. Today's viral vectors have been modified to minimize the risk of infection. The Food and Drug Administration, the FDA, has the primary responsibility for assuring drug safety. All new drugs must be carefully reviewed and approved prior to being given to humans. The FDA has assembled panels of experts in all types of new therapies, including gene therapy. New drug candidates that do not pass this rigorous review will not be allowed to enter clinical trials. The National Institutes of Health also plays an important role in ensuring the safety of gene therapy research. All studies that are supported by NIH funding must be reviewed and approved by the Office of Biotechnology Activities and the Recombinant DNA Advisory Committee to determine if it raises medical, ethical, or safety issues. Research that does not meet NIH board approvals will not be funded. Another layer of safety review is provided by institutional review boards, or IRBs, and institutional biosafety committees, the IBC. The IRB is comprised of scientific and medical advisors and consumers who review and approve all research. The study physician will also be monitoring patients during the clinical trial for any side effects. In addition, studies may have data monitoring committees, independent of the study team conducting the study, who periodically review safety data for any risks and make recommendations about the conduct of the study based on that review. Together, the FDA, the NIH, IRBs, and IBCs each perform independent reviews along the drug discovery and development pathway to ensure patient safety. Because Phase 1 clinical trials are testing new drugs never before used in humans, they carry some risk of unexpected outcomes. The FDA and multiple other organizations are there to ensure every reasonable precaution is taken to reduce risk of harm to the patient. The FDA is there to help ensure drugs perform as advertised. The FDA has the authority to stop any clinical trial based on safety data. It can take years to complete the clinical trial process and be approved. I hope you've enjoyed this overview of safety in gene therapy, and thank you for watching.